Hello everyone, I am RUBA, Assistant Professor from the Department of Nutrition and Dietetics, Trinity College for Women, Namakkal. In this presentation, now I am going to explain in detail about the oxidation of fatty acids. As we all know, fatty acids are an important source of energy for our body to function. Oxidation is the process where energy is produced by degradation of fatty acids. There are several types of fatty acid oxidation that takes place in our body: beta, gamma and alpha, of which beta oxidation is an important process, and we will see about the beta oxidation in detail. Beta oxidation is the process by which oxidation in the form of acyl-CoA molecules are broken down in mitochondria or in peroxisomes to generate acetyl-CoA, which enters the TCA cycle. I am repeating it. In beta oxidation, fatty acids in the form of acetyl-CoA molecules are finally broken down to acetyl-CoA. This is the end product of beta oxidation, which then enters the TCA cycle for further metabolic process. It occurs in mainly tissues including liver, kidney and heart, whereas it doesn't occur in the brain as fatty acids can't be taken up by the brain. In this slide, we will see about an example. There are many different types of fatty acids, of which we will take a palmitoyl-CoA as an example of fatty acid oxidation. There are three different stages in the beta oxidation, of which first is the activation of fatty acids. That is, the plain fatty acids will get activated. Only after activation it enters into the mitochondria. Second process is the transport of activated fatty acids into the mitochondria, which is called carnitine shuttle. Then third step is the proper beta oxidation process. So for the first step, activation of fatty acids, this proceeds by fatty acid thiokinase enzyme, which is present in the cytoplasm of the cell. This thiokinase enzyme requires ATP, coenzyme A, magnesium, and so on. The product of this reaction is fatty acyl-CoA. So in the activation, fatty acid in the presence of coenzyme A and ATP is converted to fatty acyl-CoA. This is the first step, and in the second step, transport of fatty acyl-CoA from cytosol into mitochondria. Long-chain acyl-CoA travels the inner mitochondrial membrane with a special transport mechanism called carnitine shuttle. Only with a special transport mechanism, the acyl-CoA will enter into the mitochondria. This is the sequence of reactions that takes place. First is in cytosol, fatty acyl-CoA combines with carnitine to form fatty acyl carnitine in the presence of acyl transferase enzyme, and in the presence of translocase it enters into the mitochondrial complex, and after entering, fatty acyl carnitine get broken down and a fatty acyl-CoA is released and carnitine will get back into the cytosol. So this is the carnitine shuttle process. Okay, then in this third step, the second step is the transfer of acyl-CoA after activation into mitochondria. What happens is that acyl groups from acyl-CoA is transferred to carnitine to form acyl-carnitine, catalyzed by the enzyme acyl transferase. Then acyl-carnitine will then shuttle across the inner mitochondrial membrane by translocase enzyme. The acyl group is transferred back to coenzyme A in matrix by carnitine acyl transferase enzyme 2. Finally is carnitine. After releasing the acyl-CoA into the mitochondrial matrix, carnitine is returned back to the cytosolic site by translocate enzyme in exchange for an incoming acyl-carnitine. The third step is the proper beta oxidation, which takes place inside the mitochondrial matrix, and this step consists of four different stages. And in the first step, oxidation takes place by FAD-linked dehydrogenase enzyme. In the second step, hydration by hydratase enzyme, and in the next, oxidation by NAD-linked dehydrogenase enzyme, and the final step is the cleavage step, that is the thiolytic cleavage by thiolase enzyme. So in the first step, that is oxidation, what happens? Acyl-CoA undergoes dehydrogenation, that is removal of hydrogen by an FAD dependent flavoenzyme acyl-CoA dehydrogenase. A double bond is formed between alpha and beta carbon, that is second and third carbon atom. In the next step, hydration takes place in the presence of enol-CoA hydratase enzyme. In hydration, where the hydration takes place, the double bond to form beta hydroxy acyl-CoA. In the third step, oxidation, that is addition of oxygen or the removal of hydrogen, takes place in the presence of the enzyme beta hydroxy acyl-CoA dehydrogenase, which is catalyzed by the second oxidation and generates NADH. The product formed is beta ketoacyl-CoA. Then the fourth step is the breakdown step, that is the cleavage. The final reaction in beta oxidation is the liberation of 2 carbon atoms, that is in the fatty acid, maybe a 16 carbon and 12 carbon. That 12 carbon atom, when the first step, in the first cycle of beta oxidation, only 2 carbon atom gets released and the acetyl-CoA then forms acyl-CoA. This occurs by a thiolytic cleavage catalyzed by beta keto-acyl-CoA thiolase. Okay. The new acyl-CoA containing 2 carbon atom less than the original re-enters the beta oxidation cycle, and this cycle repeats until the fatty acids converted to 2 carbon atoms. The process continues till the fatty acid is completely oxidized. So this is the schematic representation of the beta oxidation. First, what happens? Acyl-CoA is the first step. Acyl-CoA is converted to trans-acyl-CoA. This is the first step of beta oxidation. This takes place by the enzyme acyl-CoA dehydrogenase, and in the second step, hydration takes place by the enzyme enol-CoA hydratase to convert into beta hydroxy acyl-CoA. That is, in the beta position, hydrolysis takes place. In the third step, again dehydrogenation takes place in the presence of the enzyme beta hydroxy acyl-CoA to form beta keto-acyl-CoA, and in the fourth step, cleavage takes place and the 2 carbon atom acetyl-CoA released, which enters the TCA cycle for the further oxidation for further metabolic process. This after the cleavage, the acyl-CoA, after the 2 carbon atom gets released, the acyl-CoA again enters again the beta oxidation cycle gets repeated till the complete oxidation takes place. So in this beta oxidation, we took palmitoyl-CoA as an example. So this is the final reaction. Palmitoyl-CoA in the presence of 7 coenzyme AH plus 7 FAD molecules, coenzyme molecule, plus 7 NAD plus molecule and 7 water molecule gets oxidized to 8 acetyl-CoA plus 7 FADH2 is generated, and 7 NADH plus 7 hydrogen atoms gets released. So palmitoyl-CoA undergoes 7 cycle of beta oxidation to yield is 8 acetyl molecules. Acetyl-CoA can then enter citric acid cycle and get completely oxidized to carbon atom and hydrogen through electron transport chain. So this is the final energetics of beta oxidation of palmitoyl-CoA. In the beta oxidation of 7 cycle, 14 ATP molecules gets released, and from 7 NADH molecule, 21 ATP molecules get released. So this is from one cycle. So from 8 acetyl-CoA molecule, totally 96 ATP molecules gets generated. So some total 131 ATP molecules get generated from the acetyl-CoA was from palmitoyl-CoA molecule, of which 2 ATP molecules is used for the process. So totally 1 molecule of palmitoyl-CoA undergoes beta oxidation to release 129 ATP molecules. Hope you understood beta oxidation. Thank you for watching.